Erika. So hi everybody, thank you for being here. So as she said, my name is Erika Nava, and I'll be talking to you about the effects marine protected areas can have on fish foraging behavior. So first, I'll give you a little bit of background about myself. So I was born and raised in Watsonville, California, so up north, and it is very close to the Monterey Bay Aquarium, which is very known for its awesome aquarium and fishes that it has. So that, I think, kind of instigated a little my love for marine biology. So now I'm going to talk to you a little bit about my education. So after I finished high school, I went into my community college. Then I transferred to Cal State Monterey, and I started my scientific diving training. So I got my love for the ocean, started getting into the water, scuba diving, and then I finished my bachelor's degree in biology at Cal State Monterey Bay. And after that, I continued and got the opportunity. I got lucky enough to get a volunteer opportunity to work out here at Catalina. That was my first summer in 2015. I was out here. And during that summer, I actually was working on a goby project. So my advisor now loves to work on blue-banded gobies. We have another student out here that also will give a talk on blue-banded gobies in a couple more weeks. So if you want to learn about that, you should come to that talk. And this is just a picture of some of my previous colleagues and yes. So currently I am at Cal State Northridge. I'm working on my master's degree in biology, specifically looking at fish ecology. So I'm in the fish ecology lab under Dr. Mark Steele. So Mark Steele has worked here previously many of years and mostly works on blue-banded gobies and black-eyed gobies as well. So he has done a lot of research out here at Wrigley. So now I'm going to talk to you briefly about some of background knowledge about what I do. So here we have size-selective fishing. So if we go back into the past, we have a picture here in 1958. This cove, you can see all these fish are way larger than this little girl that you see here pictured. But then if you go back to that same location, so you take a new picture in 2007, and now the distribution and the size of the fish has completely changed. So pretty much our influence of fishing has changed the overall abundance and the fishing size that is now being captured. So because of these, the fishing pressure, we have introduced new management strategies in order to help the fish recover and maintain them in a healthy ecosystem. So here we have Fish and Wildlife. That's one of the most common management strategies, and they pretty much have set sizes of take and also size limits of the individuals that are getting captured. Another way to protect our fish, we have introduced marine protected areas. What this basically is an area where there's protection. It can vary from complete protection, which has no take of no individuals, as we have here in Fishery Cove, so no individuals are taken. And we have other areas where some take, some individuals is allowed. So here you have a picture of California, and all these little dots represent different kinds of MPAs that have different types of protection. So overall, there has been studies worldwide looking at MPAs to see if they're actually helping the fish and the inverts in that area. Here we have just a basic graph showing that looking at the biomass, the density, the size, and the richness, comparing it inside versus outside an MPA, overall MPAs have benefited the individuals within them. So we're seeing positive results because of the implementation of MPAs. So for my research, I am looking at mostly just MPAs located in Southern California. So all the MPAs that I look at are down here. So now a little bit more background. Trophic cascades is an ecological phenomenon depending on the abundance of the predator. So here we have fish, will overall influence the abundance of a prey species such as maybe seaweed. So for this example, we have a fished area, have low amounts of fish because we fished them out, and now the urchins are growing higher abundance because there's no fish to eat them. Therefore having less of it. higher impact on the seaweed, therefore reducing its abundance. But then when we introduce reserves or MPAs, we now increase the abundance of the fish, so we see higher densities of fish, therefore now controlling the urchin population and allowing the seaweed to come back. So my question is pretty much looking at, now that we have higher densities of fish in these protected areas, do we have enough food for all these fish? So pretty much looking at the inverts and the algae that's within the area. So now, in order to answer that question, I selected a study species. So I look at the California sheephead, which is common here in our waters. It has been seen from Monterey Bay all the way down to the Gulf of California. It is a journalist invertebrate. What this means is pretty much it eats a wide variety of inverts. Inverts such as urchins that are large and crabs, and eating also inverts that are smaller such as bryozoans and barnacles. This is a picture of bryozoan and an urchin above. So I also selected this species because it has a lot of studies that have been done on its diet that look at their guts and see how much it eats and what it eats. So it's a good model of species. Another good reason why I selected this species is it has actually shown that there's higher densities in the protection. So here, this was a study that was done close by here at Wrigley. I think it was intakes pipes, and this one was looking at the overall biomass of sheephead. So here in the black, you have within the protection, and in the white, you see outside the protection. You can see that overall, the biomass has increased within the protection. So therefore, in order to actually do the research and study and look into these fish and seeing if they have enough resources in the area, I have developed some different methods in order to look at this. So one of the ones I started off doing last summer, I started doing surveys. So pretty much I swam around, laid out transects, as you see here, and counted all the fish in the area in that transect, and also the inverts, so the prey species, the food availability for these individuals. After I did that, I continued to do now, if I want to know what the fish is doing, I have to watch it and see what it's eating, if it has enough food in the area. So therefore, I did time budget surveys. So pretty much what this means, I follow fish around for 5 minutes. So here I have the California sheephead, my study species. And in order to see like a non-targeted species, so rock grass is not fish, I wanted to see as more of a control to see if this fish is not being attacked or being fished has a different feeding behavior. So I did observations. So first, I had to select an individual that I was going to observe. Once I selected that individual, I started to do my observations. So I looked at their search time. So search time was described as time they're spending looking around in the dirt or in the sand looking for food, obviously. So in an area where there's less food, they're obviously going to take more time searching around for food because they can't find it. But if there is a lot of food, they're going to spend smaller amounts of time looking for food because it's very abundant. So that's pretty much what I was trying to see. I also looked at their foraging rate. What this means is just that I was looking at them, how many bites they were taking, how much food they're actually consuming. So are they eating enough? So that's one of the methodologies that I used. And I did this for around 30 individuals per site, per species, so a lot of time underwater. After this is kind of what I'm doing now this summer. So I'm doing baiting experiments. This is a baited remote underwater video. So basically here, it's a device. It has my surface float, which allows me to see it from the surface and also to pull it up. Here in the middle, we have my camera. So it's a GoPro that's attached onto there. And then here on the bait cage, I attach squid to attract the fish and see how they're reacting to the foraging, to the food that's available for them. Just to see it in a different aspect to make sure that my introduction of scuba diving is not affecting their behavior, so I introduce a camera to observe them. So here is kind of how it looks like. So I have my measuring device here, so I can kind of estimate their size. And I have my bait cage with squid, and pretty much, if everything goes as planned, we should have sheephead. attracted to it, coming close to it, and then I can size them up and estimate their size, and then seeing how much they're eating. So now here I have an actual picture how it looks like underwater. So this is actually here right around the corner on Intake's pipes. We have a lot of different fish that are attracted to. We have an eel. We even have some sheephead, which is what I expected to see. And we also have the occasional Garibaldi that's interested in seeing what is going on. So hopefully this all goes as planned. I will show you some live footage. I'm going to fast forward it. You see a lot of individuals are getting attracted to it. Some of them are like just swimming around. So here we see a Garibaldi that's getting close to bite it, but it's like, no, maybe not. So we see a lot of different things underwater. Garibaldi are very attracted to it. Easy meal, why not? Hopefully eventually you'll see a sheephead. You see a couple just swim by. There's a lot of different fish, and hopefully you'll get to see them. The tanks also. See all these different individuals that we see here in our code. Hopefully, eventually the sheephead comes by. Tail bass. Big tail bass. There's our sheephead. Up, taking a bite.
So that one would be about 20 centimeters long, or is that measuring in centimeters?
So this is measuring, this is 30 centimeters across. They get very curious, just wondering what is going on. So this measurement across is 30 centimeters.
That's pretty cool.
So right now I'm doing it all the summer. So I'm doing it at different locations at here in Catalina, most of my sites, and then I do San Diego and Kappa and Laguna Beach. I have a lot of these videos, but we will continue. So overall, my study is looking at how the MPAs affect the foraging behavior of all these different fish. So I started off with just looking at sheephead, but looking at this video, I can also look at other individuals as well. I'm also looking at, so now, some of my data right now looks like it's possibly having some research limitations towards sheephead, but I still have to continue and look into this further and seeing if this is actually a true pattern. So overall, my research kind of shows a different aspect that hasn't been really looked at. So most studies now in the past have been looking at how MPAs are very beneficial to all these fish, but what we haven't been looked at is now that these higher densities of fish are in these areas, how the fish have, if they have enough resources within them. So that's kind of the aspect that I'm taking into account. So overall, I have had a lot of help in order to do this project and continue doing this project throughout the summer. A lot of researchers have helped me, so field assistants just with scuba diving, as well as just sitting on a boat and launching my boat into the water. Also had a lot of help out here at Wrigley. I'm very fortunate to be able to come back this summer and continue my research out here. So thank you all for being here, and that's all I have for you guys. Question.
You actually seen the sheephead eating the urchins? How did they get in between those spiny spines?
So it depends on the size of the individual. So a study that has been done not too long ago was looking actually at seeing how big the urchin has to be in order for the sheephead to eat it. So the larger the sheephead, the bigger the urchin it can eat. The smaller sheephead cannot consume large urchins.
You've actually watched them do it, though?
Yeah,
have you actually seen it?
During my observations when I did follow fish around, I saw a couple times, but for the most part, they were actually not eating only urchins. They were eating stuff in the like worms in the sand, or they were eating snails from like the rocky reef. So they eat a lot of different things besides urchins. Yes.
They eat anything
Yes, so they eat a lot of, so they're generalist invertebrates, so they eat crabs, they eat brittle stars, they eat bryozoans, which are tiny. So I've seen them eat a wide variety of individuals.
There's that invasive species, is it an urchin or
So you said what, an invasive species?
yeah, do they eat the, you know what I'm talking about?
No.
I think it's an urchin, some type of urchin that eats kelp, and it's
At the root and the otters.
Okay.
When we got
So
rid of the
there, we
otters.
have had some like areas where we had a lot of urchins that kind of haven't been controlled because there's no otters in the area or there's no other fish eating them. So if there's like an area that is fish and there's less fish in that area, so therefore the fish can't eat the urchin. Therefore there's called urchin variants. So I was actually at Anacapa Island, so the backside of Anacapa is an urchin variant. So it's pretty much covered in brittle stars and urchins because it's being fished very frequently.
Talked about resources, possibility of them that are not having enough.
Mm-hmm.
So what would be what's a possible strategy to alleviate that?
Yeah, so right now, I think potentially in the future, making the MPAs a little bit larger to allow for an expansion of the fish to have more area to forage on. So using all those resources. And maybe potentially having occasionally getting fish out of there just to reduce the amount of pressure they're placing on the invertebrates. So that's another option. Question. Yes, I have actually seen a couple of different sharks. So I have been here, I've seen leopard sharks. I've seen giant sea bass. I've also seen at San Diego recently, I saw a 7-gilled shark actually attracted to my growth. So I've seen a lot of, even rays are munching on it. So a lot of different fish are really attracted to this way.
What do you want to do with your career?
I want to get a job. That's the goal. So right now, at the moment, I want to finish my master's degree, then hopefully get a job in management, conservation.
What is your idea? Expansion of
Yes.
how are you going to do that? I was in environmental studies. You're going to do that with the people who want to fish.
Yeah. So it is beneficial for the fishermen as well because when we see like these areas that are protected, and if you see it now, even now, you see fishermen fishing right outside the line of the MPA because they know that the fish are going to expand and come out because they want that larger fish. So you see the fishermen are being smart about it and coming right at the edge of the MPA to collect those larger fish that are coming outside of it. So it's beneficial also for them.
Do they have problems?
So yeah, so they have enforcement that has been like trying to protect the area, but it is hard to maintain in some areas, but they're doing the best they can.
How old is a 30-centimeter sheephead?
Ooh, 30 centimeters. I'm not quite sure the age. Wouldn't know exactly how to say that.
Okay.
What size do they get to? So what's their maturity?
So pretty much fish can grow as long as they keep on eating, so they can continue growing. I have seen Anacapa has one of the largest individuals in the protection because Anacapa is one of the oldest MPAs that has been established here in California, as well as Big Fisherman Cove. So they have massive sheephead. I saw one that looked like it was this big. It was huge. It was like not just one. It was like 10 in the same area. So it's just crazy how big they can grow. But it's usually around 4 to 5 feet.
What?
4 to 5 feet.
You spent any time at San Nicolas Island?
No, I have not. Yeah. I have a lot of sites that I have to cover, so kind of hard to get to all of them. If I had more resources and money, I would definitely go to all the islands I can.
that you showed at the beginning, there's a different time to when it was. So did what you meant by them being beneficial to that the less their fish, the longer the time they have to grow, and then the fishermen get bigger fish?
Yes, exactly. Because right now they're fishing them out sooner, so they're taking all these larger fish, and then they're growing faster, having to grow increase their size really rapidly, and it's hard for them to recover. Now they're getting that extra time. Diving. Cool. So I started at Monterey, so it's roughly around 5 years now.
Thank you so much, Erika.
Thank you.